At this point, you have been grappling with a lot of new content to understand your research, but how will you ever translate this back to your classroom? At the conclusion of this program, you will have created a classroom research project, which is the curriculum you build out around your summer research. In this presentation, I will walk you through my thinking about the content of my research when I was in RET and how I determined the content I would use as the focus of my classroom research project. Hopefully after watching this, you will have a better idea of how to create a classroom research project that is based on your currently existing curriculum and not a standalone project that you have to teach in addition to your already packed schedules. To be clear, this presentation focuses on the content portion of your research project, not necessarily the practices yet. That is to come. As you're thinking about how you will translate your research back to the classroom, there are some things you need to think about in terms of content. First of all, what content did you need to know to understand your research? That's what you guys are working on right now as you build out your concept maps. Next, you want to think about what content do you already teach that connects to the content of your research. Then, what content do you want to leave out? So what things are going to be too much for your students or not aligned with what you need to teach in your classroom? And then finally, what ancillary content is necessary to scaffold the understanding of your research for your students? That will be your classroom research project concept map that you build out. So this is my final research concept map for my RET program that I did in 2018. Just for some context, I was placed in the Penn State Cancer Institute located in Hershey, and I was working on DNA mutations as they relate to cancer. Okay, so you have built out 3 iterations of your own research concept map at this point. You should continue to add to your concept map as you start to learn new things from grad students and your mentor. However, there may be points where you need to add some additional information that you gain on your own to fill out gaps in your concept map. For example, my lab wasn't working on a specific type of cancer at the time of my RET program, but I wanted to reflect the connection of my research to cancer in my concept map. The lab I worked in focused on repeat DNA sequences in the genome and how they contribute to DNA mutations. One of the repeat DNA sequences that I worked with was a repeated DNA sequence of the bases C and A, so it's like CA, CA, CA repeating. And I found in the literature that this repeat actually contributes to an inherited type of colon cancer. So if you look towards the bottom of the concept map, so right about here, this hereditary non-polyposis colorectal cancer, also known as HNPCC, is actually caused by a repeat of the CA nucleotide bases. So even though I wasn't really working specifically on that cancer in my lab, I made that connection on my own and actually found out that my lab had been involved in a project with another researcher based on this type of cancer. And the DNA sequence I was working with in the lab at that time was part of this work. So even though I wasn't directly working on the colorectal cancer, I added that piece to my concept map. So it helped me round out some of the gaps in my understanding that I had for my project. I also did this over here with these other diseases, such as to the left here, Huntington's disease and cystic fibrosis. Those are also caused by repeat sequences of DNA. So I did add some things to my research concept map to help me understand the research that was taking place in my lab. So once I had the final research concept map, what did I do next? Well, I decided the best thing to do would be to identify the content that I already taught in my classroom. Going through my research concept map, all of the pink concepts are things that I already covered in my class in some capacity, and the turquoise concepts are new things that I had learned during my summer research. So I decided that I was going to focus in on those things that I already taught in my classroom, which would be the pink circles. After identifying the concepts that I wanted to focus on for my classroom research project, I decided I should take a look at the things that I did not want to cover in my class. All of the concepts that are grayed out are things that I decided were not relevant to my classroom research project or things that I was not going to cover in my classroom. For example, in my research, I learned about different types of enzymes that were important for the process of copying DNA in our cells. But I knew that content was going to be too specific and too deep for my students, so I decided to leave it out. So once you have identified what things you already teach and what things that you do not want to include in your classroom research project from your research, then you're going to think about how you can build out the classroom content. So how did I ultimately decide on what content to include in my classroom research project? Well, I decided to hone in on the section of my research concept map that was already rich in content that I covered in my class. So as you can see, I focused on the area of the concept map with the small box down here. So the cancer and the type of inherited colon cancer, tumor suppressor genes, proto-oncogenes, those are already things that I had taught in my classroom. So I decided to focus in on there. Then I really blew that content up, adding additional content that I also covered in my class already that would support the students' understanding of how my research was an application of this knowledge. For example, I focused on the connection of my research to cancer, but in order to understand how repeat sequences of DNA can cause cancer, the students would have to understand what DNA mutations are and that cancer is caused by mutated DNA. So you can see from my cancer bubble here, I actually built it out and included the DNA mutations and the different types of mutations and how that affects proteins and so on and so forth, to kind of really build out that part of my research concept map to include things I was going to have to cover in my classroom as well. So ultimately, I was able to really expand upon the things that I learned from the summer and think about how all of these things are going to come together and develop my classroom research project. So once I was able to decide on the content I wanted to focus on, I was able to build out additional concepts, like I said before, that I would include to scaffold my students' understanding of the research I engaged in during my RET experience. You can see that there was quite a bit more content that I added that I already taught and that connected to the research and would be necessary for the students to complete the final culminating activity. So all these things up here like gene screens, genetic testing, gel electrophoresis, those are all things that I already did in my classroom but connected back to my summer research to help the students understand how repeat sequences of DNA can cause cancer. It's important to note that this concept map is now pivoting away from what you are specifically doing in the lab. So this concept map that you're going to start building for your classroom research project is no longer focused specifically on what you are doing, but more so on what you're going to be covering with your students in the classroom. I can tell my research concept map from my classroom, I can tell my research concept map from my classroom research project, which is the one that's being shown here, from my research concept map that I did in my lab over the summer, because there's a lot more of the already existing content in the classroom. So this concept map has a ton of pink bubbles, whereas my research concept map had a lot more of the turquoise colored bubbles. So that kind of helps you differentiate. These are the things you're going to teach in your classroom, whereas your research concept map was focused more so on the content that you needed to understand in order to conduct your research. So there were some new things that I taught my students that had to do with my summer research. Things like microsatellites, which is the name for the repeat DNA sequences, and then the fact that these microsatellites actually cause some DNA structural variation. I had to teach my students about that for them to understand how these repeat sequences of DNA can cause cancer. But I really wanted to focus on integrating my project into my already existing curriculum so that it's not a standalone project. So again, you want to make sure that you're really focusing on things that you already do in the classroom and then kind of adding in the bits that you need to teach the students for them to understand what it is that you did for your summer research. So I hope that was helpful, but if you have any questions, please reach out to us. We are absolutely happy to help you think about how you're going to translate your research to the classroom.